Okay, so thank you for coming. So I'm Stephen Cornell from the University of Liverpool. Let's start by thanking Wally for the invitation. I'm not sure whether I'm here as a potential end user or as a possible source of alternative techniques, or whether I'm perhaps just the outgroup to root the tree the rest of you. But I'm a theoretical ecologist. As Wally said, I was originally a physicist, but that was quite a long time ago. So I've been looking at population biology for 15 years now. In fact, I began in population biology in Cambridge, working in the Department of Zoology. So when Wally asked me to give this talk yesterday, I gave a title which is to do with the main topics of my research, which is on modelling patterns in macroecology. But it's become apparent from talking to some of you that you'd really just like to know a little bit about ecological modelling, what kinds of models there are, and how they fit to data. So I'm going to start by just a little kind of brief couple of slides to give some examples of ecological models and some examples of fitting those to data. So when you think of ecological modelling, you've probably all seen different equations like these. So the logistic equation, Lockyer-Volterra predator-prey systems, these sorts of things. These are the kind of things which undergraduate mathematicians see, and they think of this as mathematical biology. And these were very influential models in the early 20th century, and even before then, to give us some of the ideas which are still used within ecology, like, for instance, the idea of carrying capacity, predator-prey cycles, and those sorts of things. So these kinds of models are very simple. We know how to analyze them. There's a well-developed machinery for describing the properties of these kinds of models and how they relate to the various biological properties. But they're also actually used, these kind of models are also used even now to describe real biological data. So I'll just give a brief example that I worked on a couple of years ago on parasites of rabbits. So this is a data set which was collected by someone who wanted to feed their dog. So every month they go out with a gun, and according to a certain protocol, they go and shoot a certain number of rabbits. And this is from 1977 to 2002. There are more recent data. These are every month. There's a peak which says how many rabbits he caught that year. So sometimes he caught about 50, sometimes he caught just a small number. And he also happens to be a parasitologist, so he would analyse their guts for a number of parasites, including Rufilium strigosum and Strongylus retiformis. And these here show the aggregate counts of the number of parasites amongst those rabbits that he shot every month. So we wanted to look at these data to see what kinds of patterns what kinds of processes cause these kinds of patterns within the original data. So I'll just show you actually, just to prove to you that this is data. Here's a spreadsheet to show. It's showing so strangely.
Lots of zeros in that.
There's lots of zeros. Yes. This I don't know why that's.
Can you scroll up on the left-hand side, right-hand side?
It's the well. I can. If I scroll all the way down, it's missing the title for some reason. So I'll try closing that again and see what happens. So just to show you
that window on the right-hand side, you can just see it. You drag it over.
Okay. So this is just a spreadsheet of data showing each row corresponds to an individual rabbit caught in a given year. There's something to do with where it was, what kind of vegetation there was, what food it was in the gut, whether it was male or female, and the weight in grams is turned into age using a reversible formula. And then there's data to say whether it had infection by a number of parasites, including myxomatosis and Rufilium strigosum and coccidia and such like. So these kind of data were created by Brian Berg, the dog lover. And what we wanted to do was to, I'm a rabbit hater, what we wanted to do was to understand the patterns of seasonality in the infection. So this is a graph that shows that represents the change in parasite infection throughout the year, and also amongst rabbits born in different months. So each line is reconstructed based on the rabbit's weight. So this black line here shows the parasites for those which were born in month 2, so those born in February. So those born in February aged 1 month, and so on up to, I guess, aged 8 months. And the circles are actually a geometric mean of the number of parasites, and the black lines are just simple cubic splines into those data. So you can see they're just to give some clear indication of what data are. So you can see they're fairly noisy. You can see that if you can believe these fits, then there is some kind of overall season of usual pattern that the number of parasites in a cohort of rabbits increases up to a peak and then decreases. But these different peaks change with the different cohorts we're looking at. So those which are born, in this case, in month 7, those born in July, had a much higher peak infection than those who were born in February. Okay, so what I did was actually to fit a differential equation model to these data. So this is a differential equation for the number of parasites in rabbits at age A, caught in a certain month, and for a given cohort. So actually you can see that age and time caught are related directly to cohort, so these aren't three independent things. The cohort describes when they were born, T is when they were caught, and A is the age at which they were caught. And so this model describes mortality, so this is parasites disappearing just due to straightforward mortality in that cohort. This is infection, which contains a feeding rate, which depends upon age in some assumed parametric form. A forced infection, that's basically just the abundance of parasites which are out there on pasture. And this is the immunity of the acquired immunity of the parasite. So if it has a very high acquired immunity, then this reduces the infection rate. And this, we assume a parametric form for this here. And what we do is we integrate the differential equations, and we use negative binomial errors and maximum likelihood. And these are the profiles for the predictions for the infection of the rabbits as a function of cohort and as a function of time. So you can see the similarity between these curves here and these curves here. These curves are fitted to the raw data, not to these spline fits, or indeed to the averages. So this individual corresponds to one data here. And so, quite apart from the fact that we can see that the model does manage to describe basically the pattern from those data, we use an information criterion to decide which model is the most parsimonious model which can describe those data. And that model contains a number of mechanisms. It includes a force of infection, that's saying how the speed at which the rabbits become infected, and two things which describe the seasonality in their immune response. So there's just an instantaneous immune response, which says that if I'm currently in month 3, I have the strongest immune response. If I'm currently in month 9, then I have the lowest. And there's a further one which is related to the month in which they were born. So those which were born in month 4, I guess, have the strongest immune response. Those born in month 10 have the weakest. So these are obtained from fitting. I could, if I wanted to, have some kind of confidence intervals to say more about these. But the point is that the information criterion tells us that these are effectively significant, that these if you like, are really there, according to the signal in the data. And we can interpret these things biologically. So we know that the parasite tends to die off over the winter. The parasite builds up because of the rabbits, their own infection becoming greater, and therefore the parasites produce more effective larvae on pasture. So there's a reason why this peaks later on in the season. And actually this is after the peak in the infection. And then these things too make sense because we expect that earlier in the spring, that's when the rabbit density is lowest and there's therefore the most food, and they get stressed over the year, and therefore sort of around about autumn, early autumn, late summer, that's when the rabbit populations are highest and food is available. So this is just to give an idea about how we can take an ecological model which is based on the same kind of processes as those Volterra things, but with some more biological detail. And we can fit those to data and make some biological inferences. That was just an example of that kind of thing. Okay, so apart from differential equation models, which is, as I say, is still used very much, there's also a lot of use of stochastic, individual-based, or other kinds of simulation models. And this is just an example where these were used in 2001 to model the foot and mouth disease epidemic in the UK. So many of you may well remember that there was an outbreak of this disease in February. This graph here shows the black lines are actually the data measured, I think, every week, perhaps, or every 2 days, or something of that kind. So the black points show how many new cases there were of foot and mouth. The case here corresponds to a new farm that was infected, and so it peaked around about April, and there's this very long, very stochastic tail out here. And this point here, at which there was a peak, that was actually the point at which the government implemented control procedures, which were based on the advice from the epidemiologists. I'm not saying the epidemiologists got it right, but that was the point at which this we see this downturn here. And so this is models. This is actually from stochastic simulation models, and the The red line here is what happens if they had culled the infected premises at using a different kind of procedure that was actually used. The red line is the mean of simulations, or the median actually, and these red points are just realizations. You can see they're a bit different from the actual what happens overall. These here are risk maps which were derived by Neil Ferguson using a macroecological model, so using a kind of sophisticated differential equation model which takes account of space and stochasticity. And so the point is that the models contain lots of biology. They contain something which describes the probability that one farm will infect another farm, which was parameterized from data from contact tracing from the observation of when individual infection events happened. They contain parameters which are fitted to do with how likely it is that an infectious event will happen and that will cause an infection on one farm. And also they have to use the data of the distribution of farms within the country. So this here shows individual infection events. You can see that there are more in areas where there are actually more sheep farms, I guess. That's what we see here, and up here in Cumbria. So this is kind of at the opposite end of ecological modelling. These kinds of models, simulation models, are they're liked a lot by biologists because they contain lots of things which biologists think are important. They're very hard to fit to data. It's very hard to find a measure to compare models like these to the real data and to find a way of finding the best parameters. I'm not saying that it's impossible, but it's certainly very, very difficult. Okay, so another class of models which are quite often used are quite a different sort of set of models from what I've described. So far I've been describing mechanistic models that describe births and deaths. And there's also lots of activity in ecology where instead of trying to model the population dynamics of the species, instead you say, well, okay, species tend to be in environments where they have the right kind of resources. So there are models which model the incidence of the species based on their environment. So in particular, these are actually slides from an undergraduate lecture. Sorry about that. I just had to assemble these from where I found them. So if you're interested, for instance, in what's going to happen under climate change to bird species, that's what the next slide will show, then you don't have enough information to be able to build any mechanistic model for them. But what you can do is you can look at the surveys to see where they've been seen. And then you can quantify the environment by taking a series of measurements which you think might be important, which could be something to do with how warm it is, something to do with rainfall, other kinds of things, perhaps aspects and certain kinds of vegetation. And then you can build basically a regression model to say how likely it is that a species will be in a given environment. And so this is taken from a paper by Brian Huntley from 2006. And this here shows the distribution of birds in Europe at present. So the red things are, I guess, where most species are, or sorry, where the richness is highest. So in each of these points, that corresponds to a grid cell. I'm not quite sure what size they'll be, but obviously several tens or thereabouts kilometers. And so here there are, say, 150 species in each of those grid cells. Here over here in the green, there's between 20 and 50. In the yellow, there's something like 100 or thereabouts. So this is currently how many species there are. For each species, what they've done is they've taken the atlas that says where the species has been currently observed, they've related that to the environment where it's found, and then they've taken a climate change model to say whereabouts the climate going to be in 2099, and then based on that, they can say how many species they're going to be in these different squares. So there's no population dynamics, it's all just based on the idea that you can compute the likelihood of a particular species being somewhere based on the environment. So this shows that up here in Finland, the bird watchers are going to be really, really happy. Those in Spain are going to be less happy. So as I say, this is quite a different kind of model, but depending on the kind of question that you want to answer, these kind of models can often be very, very suitable. So particularly in the context of this working group, I'm sure that in the case of soil microbiota in particular, it's actually very common to say, well, let's try to correlate different species of bacteria with different kinds of environments. And often these kinds of models can be very good. So if you have something which basically can disperse very easily, if it doesn't rely on a particular biological context, so you can model each species independently, and therefore you can say, well, if it's going to be damp and humid, then you're going to get these kinds of fungus or these kinds of bacteria. Then this kind of model might actually be the right kind of model to be doing. So as I say, this is the kind of model that I can imagine maybe has been, but certainly could be used with with metagenomic data.
What form do those types of models take?
They take the form of a logistic regression, typically. So you have, in terms of the explanatory variables, you list a series of climate variables that people all agree are important. So they can be kind of degree days, they can be how severe the winter is, those sorts of things, rainfall and such like. And then they say based on those, so they usually do some kind of model validation. So it's not just a matter of doing this blindly, but they might set this to some subset of the data and see if it's the rest of the data in some form as well.
I mean, a logistic regression is a linear model, right? And,
A kind of linear model, yeah.
And typically you want unimodal responses for...
Yes, so what you typically do is you would have your so what you want to model is the probability of occurrence as a function of your environment. And so typically what you will do is model it so there's some kind of, say, a Gaussian. So there's some transformation of this, which is then just a projection. So that's typically how you do it. So there's multiple environments and the variability. And this is actually a multidimensional variable here. Okay, so that's my very brief tour of ecological models. That's just to give just some of the scene. So mostly what I'm looking at is models in macroecology, and I suppose the most perhaps the fundamental question of biology is what explains patterns of diversity, what explains ecological diversity. So whether you're looking at plankton or tropical reefs or tropical forests, we observe lots and lots of species doing very similar things in the same place, and the question is why are they there? So the field of macroecology is basically the study of large-scale patterns of abundance. And very often in macroecological studies, what we look at is the patterns of abundance and rarity per se. So rather than saying what's the abundance of this species in particular, we say how many species are common, how many are rare, or how many species are there. So if you like, we don't treat species as being of interest in their own right. We're talking about diversity, so number of species. And I guess the same is true to some extent in metagenomic data, from what I understand. You take a measure of diversity. So the classic macroecological pattern is the species area curve. This is from a paper by Preston in 1960, who was an engineer who also was a bird watcher. And these two lines correspond to species area curves for areas which correspond roughly to South America and North America, but not quite that. So what they are is, each point here is at a given scale, so this is log area in acres. How many species, on average, are there in samples of this particular area? So in Colorado, Colorado Island in Panama, that'll be. So in about 10,000 hectares, there are typically each of the 10 to the 2, so about 100 species. So what we can see is that there's some kind of triphasic curve like this. There's a broad, on these log axes, there's this broad, roughly parallel region, and then there's steep species area relationships at very large scales, because then comparing different areas of that size are typically statistically independent, and so therefore number of species is proportional to area. Just doubling the area means that you have twice as many species. And down here, this is just some kind of sampling effect at very small scales. And if we fit a straight line to this log plot, we get a power relationship between species and area. Species are like areas of power, about 0.2. Is this kind of thing familiar? Do people know this? This is kind of thing that you might see on. Okay.
Has anyone ever done that for microecological?
I think they have, yes.
Yeah, there's been quite a lot of. I mean, the one I remember is with oil sumps from factories, right? So Tom Bell does a lot of it.
Yes. So I thought it was from oil tankers. Is that right?
No, it was actually it was lathes in factories. So each lathe has an oil sump where microbes were living, and you can treat them as little islands. And then he did a species area curve on the microbes in lathes.
Well, if they're islands, how do you get a...
Well, that would be, I mean, you'd tolerate the size of the sump, I guess. That was what yeah. But they have been looked at. Generally, I think the intuition is that they're flatter, so the Z value is smaller than it is for macroscopic organisms.
I think it's been done on tree holes.
Yeah, that was common Tom
yeah,
as well.
it was, yeah.
So you can do this on the beach. You can go to look at rock pools, and you can see rock pools of different size and count the number of species of things in there. So they're very easy to reproduce.
But I think Tom Curtis may have shown that it doesn't apply to soil. Do you know Tom Curtis?
Yeah, quite well, yeah.
I think. On the
Why? Because you just take a very small amount.
because it's so vast, because you never get anywhere near the same sort of. I mean, I may be.
But that was pre metagenomics.
It was, yes. It was just modelling.
That was just clone libraries, and that was ridiculous. Yeah, but I think you can do it now. Actually, Claire Horner Divine has done lots of stuff with it, so she's one of the best. Yeah.
Okay, so here's another kind of macroecological pattern, and this is a species abundance distribution. So this is taken from Barra Colorado Island in Panama. It was actually originally not an island, but it was became an island when they flooded the Panama Canal. And there's been a well-established 50-hectare plot on this island since the 80s, where they do a complete census of all the trees. So there's hundreds of thousands of stems greater than 1 centimetre wide. Typically, we only class those as being bigger than 10 centimetres wide at breast height, that we count as being adults. So there's the order of a few hundred species, again depending on where you look and what your lowest limits of sizes that you look at. And so this is a classic way of looking at species abundance distributions. These here are abundance classes. So these are actually logarithmic classes. So the first one is how many species have abundance 1. The second is how many has abundance 2 or 3. Then 4, 5, 6, 7, 8 to 15, and such like. 9 to 8, 15, 16 to 31, and such like. And so out here, this here corresponds to those abundances greater than about 1,000, and it's a small number, 2 or 3. Down here, there are something like 25 species which have only one individual within this large hectare plot. So it's a very diverse area. And so these kinds of histograms of species abundance distribution, these are also things which are often surveyed in ecology and which people pontificate about what those distributions mean. It's actually a little more complex, and I'm not going to go through all of this graph, but these are the species abundance distribution using the same data, but measured at different scales. So down here, instead of using the full 50 hectares, I look at 0.05 hectares, and in that case I have many replicates of samples of that size, and I get a different shape species abundance distribution there. Blue curves are actually obtained by taking random samples from the total data of the same size, and the point of blue curves is just to show that the samples at a smaller scale are not just random samples from the full species abundance data. Anyway, so this is another macroecological pattern. Okay, so what kinds of processes give rise to community of diversity? Well, there are broadly three kinds, I'd say. So the reason why species can, why communities can be diverse can be either because species interact differently with their environment, so the different species have different preferences, and therefore they're able to exploit different resources within their environments. It can be that the species have different kinds of biotic interactions, so they can create their own local context, which is beneficial for them, and because many different species can do different things, this allows them to coexist. But also it could just be that the reason why there's lots of species in one place is through random processes. It can be just that there's many dispersing seeds from trees, for instance, they just happen to fall there, and just by chance you end up with lots of species because lots of species happen to arise there. So whatever we see within a given community is just some kind of remnant of population dynamics based on a series of colonization events at some point. So depending on which one of these processes we think is the most important, we build different kinds of models. So it's actually quite tricky to build a model. Well, in principle, it's not difficult to write down models which contain all these different things, but the number of parameters makes them rather infeasible. So what we tend to do is to write down a simplified model which contains perhaps only 1 or 2 of these kinds of process, and then try to see what they predict in order to say what's likely to be the most important processes going on. So it's quite clear that Charles Darwin was a big advocate of the idea that the reason why species coexist is because they're doing different things. So in The Origin of Species, there's a number of famous quotes where he talks about a tangle bank with lots and lots of species there, and he says people might think that the reason why we have all these different species there is because of chance, but actually he says it's because of very precise processes going on. And at the other end of the spectrum is neutral theory, which was proposed by Steve Hubble, who put forward a mathematical theory that says that all individuals are ecologically equivalent, so any patterns that we see are just the result of demographic statistics. So So if you have some species which are rare and some which are common, that's just because of ecological drift. And the point is that these kinds of theories, once we formulate these theories in terms of a mathematical model, we can then test these theories against patterns such as the species abundance distribution or species area curve or other kinds of patterns, and see if those models are adequate to describe those data, or whether the data show that those models are wrong. So Chris actually gave a very nice introduction to neutral theory, but I'll remind you what neutral theory consists of. So it's a model that's based on birth, death, and immigration. So it's models individuals of different species, and it assumes that all species have the same parameters. So when I say they're all the same, it's not that they are in the model you keep track of the different species, but they're all assumed to have the same birth, death, and immigration rates. And also the individuals compete for space. So this is appropriate for a tropical forest, as I was saying yesterday. So in a tropical forest, light is a limiting resource. And so individuals of different species compete for space. And so if they're doing that on an equal footing, then we expect them to compete equally for space. So if like there's a vacant gap in the canopy can be filled in principle by any species. And so with that assumption in the model, we can assume the competition between individuals is independent of species identity. So that's a fundamental assumption of the model, and we can see where that leads us. And then because all the species are, all individuals have an equal likelihood of giving birth at any one time, any patterns in abundance we see are generated by stochastic variation. So I'll just give a quick cartoon like Chris's blackboard sketch yesterday. So here's a community of 16 trees. There's 9 dark and 7 light green trees. Here's a gap in the canopy which arises because of whatever reason, termites have eaten this tree or this storm or something. And then within the model we assume that this gap will be filled very rapidly by one of the offspring of the original community. So there's seeds in a seed bank. And so if they have an equal likelihood of colonizing this, then with a chance, probability 9/16, it'll be a dark green tree, and 7/16, it'll be a light green tree. Except that if we just assume this and we follow the dynamics over time, this will then drift towards fixation. So to maintain diversity, we have to assume that there's a probability M of it being something different. So this could be either an immigrant from outside, or it could be a brand new species due to speciation. And then these probabilities are modified by factor of -n. And there we go. So that's basically what neutral theory does. And there's different versions of it depending on whether we assume that these are entirely novel species, or whether these are species which are arriving. There's a constant trickle from some other larger community. Okay, so the reason why neutral theory is taken seriously by ecologists is because it's very good at describing species abundance patterns. So here are the bars are data from either coral reef communities or tropical forests. This is actually that Colorado Island data that I described before. And the lines are the fitted neutral model. The neutral model has, I think, 3 parameters in this case, and you can fit the model very well to describe these patterns. So this is originally very surprising because people usually fitted these data with much more complex models with more parameters. So this is really the reason why neutral theory is.
This model has 1 or 2 parameters.
I have to be specific. So I guess it has 1 parameter, which is the biodiversity parameter theta. It has a third parameter, which is the rate of immigration from outside. So so it basically assumes there's a meta-community somewhere described by this biodiversity parameter, and then it has an immigration parameter M. So I think these are probably 2 parameter fits. Okay, so neutral theory is good because it agrees with data on species abundance distributions. It's also good because it's a mechanistic model. It's not a kind of a heuristic model where we say, for instance, let's model diversity as a broken stick model, which was done by Fisher originally, I guess. Maybe not. But there's lots of kind of rather nebulous models about communities saying that they're dividing resources amongst each other in some particular way. This is an explicit model that says it's births and deaths. So in principle, you can go out and measure births and death rates and dispersal and speciation and compare it with your model. It's also mathematically equivalent to neutral theory in population genetics. There are some minor differences in the sense that we have this meta-community, we have dispersal limitation and such like. So these are very minor things, but basically all the mathematics of neutral theory in population genetics carries over. And associated with this is the fact that there are statistically rigorous methods for comparing the model with data. So the model gives you an explicit algorithm to compute the probability of getting a sample containing a certain vector of abundances. So we have a full likelihood function, and and it's rather expensive to compute, but in principle it can be done. So ecologists like Kevin Gaston, who's no natural friend of neutral theory, acknowledge that it has very broad scope, and that there's no kind of equivalently ambitious theory within model within niche theory. But neutral theory also has a number of weaknesses. So although we can get it to agree with data when we fit it, if we then try to interpret those parameters, we often find that the parameters look rather unrealistic. So in particular, we find that the speciation rates and deliberation rates, which are the things which maintain validity, have to be much higher than we think independently they ought to be. So for instance, typically we find that speciation rates probably have to be the order of 1 new species per 10 to the 5 new births, which is quite a lot. That's it. So we probably believe that that's true. It's a non-spatial model, so it basically just assumes that you have this complete mixing between the individuals. So any individual can be particularly likely to be the parent of another individual. It's ultimately maintains diversity by a speciation process, and it assumes a point speciation process. So just that new species arise as a completely random process, a custom process. And of course, it has this neutrality assumption itself. So ecologists have been arguing about how different species are and why these differences are really important. And the assumption that, in fact, species are all doing exactly the same thing is really rather unpalatable. So there's no question that neutral theory is not absolutely true. So it's obviously false at some level. But on the other hand, there's no question that it can also serve as a reference point for detecting non-neutrality. So if you want to test for our particular interesting biological process, then we have to take some kind of null model to tell us whether there really is a secret in our data, or whether actually what we're seeing is just a fluctuation which looks like a pattern and really isn't. But also attached to the fact that it's very mathematically tractable, and there's very powerful techniques for neutral theory, it can serve as a starting point for more realistic models which can still retain some of the tractability of neutral theory. And that's largely what I've been doing in my group over the past few years. So I'm not going to go through all the slides which I have here, but we've been looking at ways of relaxing these different assumptions. So making the theory spatial, changing the speciation process, and also relaxing neutrality so that the model is weakly non-neutral. And what we find generally is when we relax these things, we end up with a model which has some tractability. It's not as solvable as neutral theory, but when we try to fit those models to data, we find that the parameters are more realistic. So we find speciation rates which are much closer to what we expect, and so on. So I'm not going to go into so much about the spatial neutral theory, though I have lots of slides on that. This, by the way, is what was done by James Rosendale as part of his PhD. We continue to work on this since he finished the PhD. I'll just cut the chase straight away. So the difference between spatial neutral theory and non-spatial neutral theory is that when we have a gap in the canopy, the spatial neutral theory